Ever wondered why a seemingly simple fever might make you feel like you're climbing a never-ending staircase, struggling with waves of nausea and the constant discomfort of abdominal pain? This could be the notorious enteric fever, making its unwelcome presence felt. Enteric fever is a multi-system illness presenting with a range of symptoms such as a step-ladder fever that progressively increases, vomiting, abdominal pain, diarrhea, and a rash. It's caused by the Salmonella typhi and Paratyphi A, B, C bacteria, which sneak into our bodies through contaminated food and water. So, how does this illness take hold in our bodies? Picture this. The Salmonella bacteria enter through contaminated food or water. Once they're inside, they cause the formation of ruffles on the cell membrane of intestinal mucosal cells. They then get internalized into vacuoles of these mucosal cells, and macrophages try to phagocytose the salmonella-containing vacuoles. But due to the LPS alteration of salmonella, they resist this elimination. From there, they travel via lymph nodes, getting disseminated to the reticuloendothelial system, and then via blood to all parts of the body. Diagnosis of enteric fever involves several steps. Firstly, a Gram stain reveals Gram-negative bacilli motile with Peritrichus flagella. Blood cultures are then performed in the first week of illness, using samples of blood or bone marrow culture. On a blood agar, these bacteria are non-hemolytic. On McConkey agar, they are non-lactose fermenters, producing translucent and pale colonies. Selective media such as XLD agar, DCA agar, SSA agar, and WB agar can also be used, along with enrichment media like selenite F broth and tetrathionate broth. Antibody detection tests, such as the Widal test, are performed after the first week of illness. Salmonella has somatic O antigen, flagellar H antigen, and capsular Vi antigen. Antibodies against O and H antigens are used for diagnosis. O antibodies appear early and disappear early, forming granular chalky clumps, while H antibodies appear late and disappear late, forming loose, fluffy clumps. Stool cultures are done in the 3rd to 4th week of illness, and biochemical tests for salmonella show it to be catalase positive and oxidase negative, indole and urease negative. Finally, an antimicrobial susceptibility test is performed. The clinical features of enteric fever are a fever of the stepladder type, anorexia, abdominal pain, vomiting, diarrhea, rashes, and cough. In severe cases, it can lead to GI perforation and bleeding. So there you have it. A comprehensive look at enteric fever, its pathogenesis, diagnosis, and clinical features. A seemingly simple fever, but with a complex process behind it. Stay informed, stay healthy.